Hi, everyone. We're excited to tell you a little bit about Sensei Biotherapeutics today. I'm John Chellaby. I'm the CEO of Sensei. I've got a 25-year career in biopharma. I started at the research bench, like so many people, at Telluric Incorporated. I was lucky early in my career to discover a drug that made it into human clinical trials. And I've been doing that ever since, over the last 25 years, mostly in oncology companies. I've spent time in both public and privately held companies. I have worked at both big companies like Biogen, Agilent Technologies, and Arcuel, and over the last 10 years in more entrepreneurial roles. My last role, I was Chief Operating Officer at X4 Pharmaceuticals in Cambridge. I joined Sensei Biotherapeutics around 3 years ago because I thought it was a great opportunity to build a truly differentiated and potentially transformational platform, and to build a really world-class team. I'll let my colleagues introduce themselves.
Hi there, I'm Marie-Louise Fjellskog, and I'm the CMO at Sensei. I'm a medical oncologist by training, and I got my MD, PhD, and later on my postdoc at Uppsala University and Karolinska Institute in Stockholm, Sweden. Most of my career I've been working with patients, so I've been treating cancer patients, participated in academic studies, and also ran my own research. I've always been focused on individualized therapy. I've seen the side effects patients can get from treatment, and I think it's really important that every patient gets the treatment they need, no more, no less. Every patient is different, and their tumor as well. About 10 years ago, I joined pharma. I started working for Novartis, and then I moved to Infinity, and then on to Merus. In all of these different companies, I've been the global lead for several immunotherapy combination studies. So what we have done is that we've added small molecules, antibodies, or bispecifics to PD-1 backbone. And we wanted to enhance the efficacy of PD-1 blockade in those indications that do not respond to single-agent PD-1 blockade. One thing that I always missed in my combination studies is the vaccination approach. I'm really happy that at Sensei, I can use vaccination in combination with other immunotherapies. Thank you.
Hi, I'm Rob Pierce. I'm the Chief Scientific Officer at Sensei. Like Marie-Louise, I'm a physician scientist, actually a pathologist by training, but my entire career has been spent in immunology, and in particular, trying to understand how different tissue and tumor microenvironments inhibit T-cell responses. That question led me from academics to industry, and then back to academics, and now to Sensei. At one point, after the University of Rochester, I moved to what became Merck. And that put me at the right place in the right time to help the early pembrolizumab program move through preclinical and then early clinical development. I was the medical lead on pembrolizumab in Merkel Cell Carcinoma, which is a tumor that we'll touch on later in our presentation, as well as cutaneous T. But most importantly, I ran a group that focused on trying to understand who would respond and who wouldn't respond to anti-PD-1 blockade. My group developed the companion diagnostic for pembrolizumab. And with that suite of tools, we began to see that there was this patient population that we now call patients who have cold tumors. That means they don't have tumor-specific T cells in the tumor. These patients don't respond to PD-1. And I was very fixated on trying to find a solution to that problem. That led me to leave Merck in 2013 and go to a small company called Oncosec Medical that had an interesting play in this space. And then ultimately back to academics at the Fred Hutchinson Cancer Research Center, where again, I wanted to focus on how do we reboot the immune system and give these patients with cold tumors an opportunity to respond to anti-PD-1 and other T-cell checkpoint therapies. That's when I met John, who introduced me to Sensei technology. And I really fell in love with the ImmunoPhage platform that we're going to talk to you about today.
Hi, my name is Anu Hoey. I'm the Chief Business Officer at Sensei. I have over 20 years of experience in the biotech industry and spent the last decade primarily working at companies with differentiated immuno-oncology platforms. I began my career after a graduate degree in molecular virology and spent many years doing drug discovery research in biotech. And then in the last 20 years, have worked in business development and corporate strategy at various companies. I've transacted multiple deals with total values over $1 billion, as well as spin-out companies and other licensing deals. And I will be leading business development and corporate strategy at Sensei. Thank you.
I'm James Pyer. I'm the chairman of the board at Sensei, and I've had the pleasure of working with John and his team to do the last couple of private rounds of investment in the company, and have also played a little bit of a role in helping to enable this team to go achieve their vision for the future of cancer vaccines. My background is as an immunologist, so I got a PhD in stem cell biology and host pathogen interactions before spending a fair amount of time in the investment world over the last decade or so. But my history with cancer vaccines starts from a somewhat personal story that led me to Sensei, which is that when I was a graduate student just starting my career in academia, my dad was diagnosed with cancer. He got stage 3 melanoma. And when trying to think about what to do to treat him, we got him enrolled in one of the earliest cancer vaccine clinical trials at the National Institutes of Health in Maryland. And we would fly out every few weeks to go do his treatment. And he actually responded and is thankfully still alive today because of that treatment. Actually, at the time of recording this message, of all things. So I'm so happy to have him around. But that really, that treatment that he got, he was one of only about 5% of patients to respond to it. And so I've been thinking for the last decade, what are the elements of a therapeutic cancer vaccine that would be required to get more patients to respond like my dad did? And it wasn't until I met John and this team at Sensei that all of the elements of a great therapeutic cancer vaccine were finally united in a single platform for me. And that's what I think the team is going to tell you all a little bit more about today. So thanks for being here.
Thanks, James. So I think if we start on slide number 5, you'll get a pretty good feel for the 5 strategic underpinnings of what makes Sensei. And I think it starts and is really centered around our proprietary ImmunoPhage platform technology. It's probably worth spending a minute just talking a little bit about corporate history so you fully understand how this technology evolved. The company was actually started in the year 2000, so it's about 21 years old, and it spent about 15 years in stealth mode. And I don't believe the company was ever started to bring a drug into clinical trials or to become a publicly traded company. To be honest, it was really more of a retirement project for the founder, who had spent a long career at Abbott Laboratories and wanted to pursue some interesting scientific ideas. And over the course of that time, he raised a little bit of money here and there. And the technology evolved to the point where by 2015, it was ready for a clinical study. And so the company raised its first really meaningful investment and ran that phase 1 study, began to read out in 2017, and it looked really, really interesting and promising. And so the board said at the time, let's bring in a oncology-focused team and really bring this company to the next level. So I joined in 2018. Robin Marie-Louise joined not long after. James and Christian Ungermeyer became our lead investors around 2019, and Anu joined us last year. And the reason I mention all that is because it really wasn't until 2019 that the first academic paper published showing that you could shrink tumors in a HER2-driven mouse model using a similar type of approach. So we feel like we have a good 4 or 5 year head start over the rest of the biotech world. From that platform technology, we built a pipeline of cancer-focused immunotherapies. The most advanced asset is in a phase 2 study now in head and neck cancer, and those results have begun reading out and also look very promising. That's why we're here today. We also have built collaborations with AstraZeneca, Adimab, and other academic institutions focused in oncology. And we built our own manufacturing facility. And the reason I bring that up is because we integrate manufacturing into the earliest part of our discovery effort. It's an important part of how we plan to transform personalized medicine. And we'll tell you a little bit more about how we're doing that as we go through this presentation. Now on slide number 6, you'll see a view of our pipeline. And as you can see, SNS-301 is our most advanced asset. It's an immunophage targeted towards a tumor-associated antigen called ASPH. That's an antigen commonly found on tumors, but much, much harder to find on normal healthy tissues. And very soon, this is going to be an immunophage cocktail consisting of HPV antigens as well, because about 70% of all head and neck cancers are driven by the human papillomavirus. Right now, we are enrolling in a phase 2 study in first-line head and neck cancer. So that's the first line of treatment after surgery in combination with pembrolizumab, a drug that Rob was one of the early leaders on. Very soon, we'll be starting a second study in collaboration with AstraZeneca, and this will be in the neoadjuvant head and neck cancer population. And neoadjuvant patients are those that are treated prior to surgery. That's a very large population of patients and also quite a bit of unmet need. The second program is SNS-401, and that's a really critical program for the company because this is a microcosm of how we intend to deploy our platform in the future. SNS-401 is a cocktail of ImmunoPhage targeted for a group of patients called Merkel Cell Carcinoma. That's a type of skin cancer. And it's a very interesting type of cancer because it's virally driven. And in collaboration with the University of Washington, we're deploying some proprietary approaches to develop this cocktail. And Rob will talk to you a little bit more about that. And then the third program is an antibody program targeted towards an immune checkpoint called VISTA. We believe strongly in this target because VISTA is an immune checkpoint that is quite different from the PD-1 pathway, which is where most immune checkpoint drugs are pointed towards. But VISTA is interesting because it plays a role in the resistance to PD-1 inhibitors. So we think it's highly complementary to our efforts in the ImmunoPhage platform as well. Now, if you look at slide number 8, I think it's useful just to spend a moment before we tell you where we're going to tell you where the industry has been in terms of cancer vaccines. Because if you rewind the clock 15 or 20 years, you would have seen a large number of studies ongoing for cancer vaccine drugs. And these were really developed at a time when the mechanisms of resistance were not that well understood. And these were first generation approaches. So unfortunately, most of these drugs were not approved. There just weren't enough patients like James's dad to reach the levels of response that were needed to get FDA approvals. And we've learned a lot since that time. And one of the key modalities that's really made some strides is in the area of checkpoint resistance. We almost take them for granted today because they are so successful. There are 7 of them approved. They're approved in over a dozen different cancer indications, and it's a $20 billion market that's set to double over the next several years. Unfortunately, though, these drugs still only work in a minority of patients. In head and neck cancer, where we're running our phase 2 study, the objective response rate on Keytruda is 18%. That's 18%. The progression-free survival time is 2 months. The median overall survival time is 8 months. So there's a lot of work to do that remains. Now, Rob and Marie-Louise have spent a good portion of their career working on the reasons why some patients respond and some patients don't respond to checkpoint inhibitors. And today we understand that there's really 2 mechanisms. The first is you can think of checkpoint inhibition as something of a brake on the immune system. And it's a brake that tumors deploy in order to avoid destruction by the immune system. So when you treat with a checkpoint inhibitor, you're releasing that brake. But if there's no immune system around the tumor, releasing the brake will do no good. You actually have to drive the immune system to the tumor, around it, and so it can infiltrate it for checkpoint inhibition to work. The second reason, as we now know, is that sometimes there are multiple brakes. Just like on a car, there's an emergency brake. In cancer, sometimes there's 3 or 4. So we believe, and the central question we're asking with our ImmunoPhage platform is outlined on slide number 9, that if you target both of those key mechanisms of checkpoint resistance, you have a lot more chance of being successful. And that's really the primary question we're asking is, can we engineer a virus to target both of those key mechanisms? The second very important question is, can we transform personalized medicine using this approach? Personalized medicine is absolutely the right way to treat cancer patients. If you took 1,000 biopsies from 1,000 different patient tumors and you genetically sequenced them, I can guarantee you they would all have very unique genetic profiles. So it's the right approach. The problem is that current approaches to treating those in a personalized way take too long and they cost too much. I've already told you some of the dismal statistics in head and neck cancer, and that is really illustrated on slide number 10. And that's because of the use of bacteriophage. Bacteriophage are unique viruses. They infect bacteria, but they don't infect human tissue. They don't infect normal healthy tissue, mammalian tissue. But perhaps more importantly, they are highly immunogenic to the immune system. And Rob's going to tell you a little bit more about why that is. They also have very substantial genetic payload capacity. So we can genetically program them in ways that both drive the immune system to the tumor in a focused way and also releases the brake on the immune system. We think we're the only company doing both of those mechanisms with one therapeutic product. I think most cancer companies are either targeting that first mechanism or the second, but not both. The third thing that's really different about ImmunoPhage is that they're incredibly fast and efficient to manufacture. So that's part of the reason why we think bacteriophage are an important contribution to cancer immunotherapy. Now, Rob's going to tell you a little bit more about how we do what we do, starting on slide number 11.
Thanks, John. You know, when I was at the Hutch prior to joining Sensei, I spent some time thinking about the problem of why cancer vaccines hadn't sort of delivered on their promise. And John has gone into one aspect of that, which is the presence of T cell checkpoints, which can block the activity of anti-cancer T cells if they get into the tumor. But there's a flip side that I want to focus on, which is rebooting the immune system to drive the development of those cancer-specific T cells. And that's critically important. And so when I looked under the hood at the ImmunoPhage technology, I fell in love with it and got really excited because I thought that really we had here a platform where you could check all the boxes that really I thought were required to drive those T cell immune responses against cancer. And what were those boxes, and why was I excited? Well, I think that to really have an effective anti-cancer vaccine, you need to maximally mimic a pathogenic virus, something like we see with acute viral infections like hepatitis A or yellow fever, where we mount a strong, deep, what I call a complete immune response. That's not a technical term, but what I really mean with that phrase is that we're getting strong innate immune activation and then the downstream activation of adaptive immunity, meaning B cells and antibody responses as well as T cells, and the T cell component includes both CD4 and, importantly for cancer, CD8 T cells, which are the foot soldiers of the immune system that actually then go in and kill the tumor. So with the bacteriophage, I mean, the brilliance of this, and that I can't take responsibility for, was that we could take bacteriophage, which has been used for decades as what we call phage display, and that's a tool in the lab. But we had already had a platform that was extremely easily engineered. We can put large proteins on the capsid, we could manipulate the genome of it, and this was sort of ready to hand. But the founder of Sensei realized you could flip it on its side, and it was already a strong immunostimulatory vaccine platform, and that's what SNS-301 is doing. But I want to dive into a couple of deeper features here. So we put antigen as fusion proteins onto the capsid of the virus, and we could put big pieces of protein, not just small single epitopes. I think that's important for a variety of reasons that we'll get into later. But so we could put lots of antigen and big pieces of antigen on the surface. But what's more, the ImmunoPhage is naturally capable of stimulating the immune response, and that's in part due to the many layers of danger signals as well, because the bacteriophage, though it does not infect our cells, is in fact a virus, and the immune system reads it as a potential danger and reacts accordingly. And what that does is captured in the cartoon number 2 here, which is to really drive an effective immune response, and in particular to T cells. There are 3 critical signals that the antigen presenting cell, now this is the cell that's sort of the sentinel of the immune system, that encounters the antigen, takes it up. The dendritic cell has to sort of deliver the appropriate signals downstream to T cells. Now, the magic here really is, the secret sauce is, we trick the immune system into thinking that this bacteriophage, which looks dangerous, is a cancer cell because we code it in antigens. These are the pieces of protein that the immune system sees as foreign and attacks. So if you can see in this cartoon in number 2, the phage is taken up by the dendritic cell. The dendritic cell chops up all of those proteins and then presents antigens to the T cell. And this is really the what. It's telling the T cell what to focus on attacking. It's like, this looks like a dangerous virus, but in fact, it is now a piece of cancer antigen that we're displaying to the T cell. So that's what to attack. Signal 2 is called co-stimulation. And what it really is asking the question, really? Should we really attack this? Is it really dangerous? And it's often likened to turning the two keys in a missile silo to allow for launch. You need to turn signal 1 and signal 2 simultaneously to get the T cell to go, and that's really important. Signal 3 are the cytokines, the molecular messengers that the dendritic cell puts out into the local environment in this communication with the T cell to really tell the T cell which kind of immune response are we going to make. Are we going to attack? Are we attacking a parasite here? Are we attacking a virus? Are we attacking cancer? And really, we know that IL-12 and interferon gamma are very important cytokines that help tell the immune system what flavor of immune response to generate, and also help to contribute to the fitness of especially CD8 T cells to be cancer-killing cytotoxic T cells. So on the next slide, slide 12, or actually, James, do you want to jump in? Because you usually do a great job elaborating on, especially on these 3 signals.
The only thing that I would emphasize is that these 3 signals, signal 1, 2, and 3, happening at the right place and at the right time is really what we look for a great cancer vaccine. And that's when I came in to partner up with you all on this, that was what I was looking for. And I think that's what a big part of what makes Sensei so unique is that I haven't seen another vaccine platform that can deliver 1, 2, and 3, all the signals in the same place in the same time to a T cell.
On slide 12, I would just like to point out, this is an experiment that my postdoc did it before we left the hutch, asking the question, is ImmunoPhage really intrinsically immunostimulatory in the way that we just showed on the previous slide? What we did was we took SNS-301, this is our current clinical phage that's in phase 2 in head and neck cancer. It is a immunophage that is expressing a large piece of an antigenic fragment of this antigen, ASPH. But we're really focusing here on that innate immune activation. And so in the clinic, we do intradermal injection of SNS-301. And here what we did is we took normal skin from reconstructive surgeries, and we injected SNS-301 into it. And you can see on the top row, you see a really nice elaboration. This is by flow cytometric analysis. You can see that, yes, in fact, SNS-301 is driving signal 2. CD80, CD86, and CD40, which is really important for licensing the generation of CD8 T cells, all go up strongly in a dose-dependent manner. So that was really wonderful to see. Signal 3 is cytokines that are elaborated and secreted into the tissue culture medium. You see a bevy of different cytokines indicating, again, that we're getting a strong intrinsic immunostimulatory reaction when the phage interacts with dendritic cells. So on the next slide, so that's the previous slides focused on our current state of ImmunoPhage. One of the things that really excited me about this platform was not only was it sort of the base case, strongly immunostimulatory, but the fact that this system is so easily genetically engineered allows us to actually augment it even further and specify the kind of immune reactions that we'd like to see. And we did this by really a marriage of 2 really cool orthogonal technologies. And I'll start over here on cartoon on box number 3, which shows an alpaca. So when I was at the hutch, I was a founder of a small biotech called Avaxa Biosciences, where we focused on making alpaca-derived nanobodies. Now, you may have followed the Ablig story and you know about nanobodies. Long story short, these are antibodies that have many of the same features as a full-length antibody, but they're small antibody-like fragments at about 1/10 the size of a normal immunoglobulin molecule. What that allows you to do is make inhibitory antibodies or immunomodulatory antibodies that are much smaller and are encoded by a much smaller piece of DNA. So if you're working with a system, which at the time was a lentiviral CAR-T system that when we were at the hutch, we were trying to figure out how to pack the most immunomodulatory punch into a restricted payload. And so hence we went with the alpaca-derived nanobodies. And you can see in the bottom right are some of the targets that we were prosecuting at Alpaca. When I was talking to John about coming on as the chief scientific officer of Sensei, we looked at these Alvaxa assets and we realized that all of these molecules that we were working on could be easily ported over to the ImmunoPhage platform as well. And so that's depicted here in box 1. So we could put antibodies or nanobodies on the surface of the phage to help direct the phage to specific cell populations, and when they get to that cell, to activate those cells. And that's depicted in the blue T's there. But what's more, John mentioned the payload capacity of bacteriophage. It's not well known about bacteriophage, but in fact, bacteriophage are a decent gene delivery vehicle. And we have a rather large payload space where we could encode within the bacteriophage things like immunomodulatory nanobodies, so that when the phage gets taken up by a cell, it begins to make those nanobodies that are encoded within the phage. And in this way, we have multiple different means of both enhancing the targeting, but also enhancing the effect of the immunophage on those target cells. So we're really excited about these two technologies coming together in our future embodiments of immunophage. Now on the next slide, I want to focus on something we've already touched on. This is slide 14. So phage enables us to do a couple of critical things. And really it's because of the manufacturing process that John alluded to. We can make them cheap, we can make them quickly, and what's more is they have really long shelf life. What's more, I think I mentioned earlier, we can put rather large antigenic domains, and we've put up to about a 400 amino acid piece of antigen onto the surface of the phage. What that allows us to do is we have made a bank or a library of immunophage that express very common shared tumor-associated antigens across all different tumor types, right? So we have this bank already made, and we're continuing to deposit more phage into that bank. And what that will enable is that we can treat a future Sensei patient, and this is captured on slide 15, in the following manner. We get a biopsy, we do a quick analysis of the expression of shared tumor-associated antigens, the ones that are in our bank. When we have that data, maybe a patient has 5, maybe a patient has 10 of these shared antigens, we can reach into our GMP drug substance that we've already pre-made, make a bespoke cocktail formulation of it, and ship it to the site in about a week. So we can already begin treating a patient with a personalized cocktail very quickly. And that addresses the problem that many personalized approaches have in terms of it takes so long to get the therapy to the patient. Here we can begin treatment probably in about a week. What's more is we can then dive down deeper into a mutational analysis of that patient's tumor and pull out candidate mutation-derived patient-specific neoantigens. And because of the speed of manufacturing of our phage, we can put these neoantigen candidates on a phage or 2 phages and cover almost, up to probably 50 of these candidate neoantigens, and then ship that to the site and treat the patient probably 4 weeks later with a booster vaccine that includes those initial shared antigens as well as these mutation-derived patient-specific neoantigens. And we really feel this hybrid approach differentiates us from most others in the field.
This ability to do this with the ImmunoPhage platform and the library that Sensei is building, I think it's really marrying the revolution that we're seeing in genomics and genetic sequencing, right? The ability to routinely sequence every gene that's expressed in a tumor and every mutation that it has with therapeutic decisions that physicians can make, giving, kind of the cart and the horse over to doctors to create a personalized therapy.
Now, before we hand it off to Mary Louise to take you through some of our clinical data, I just want to spend 1 second on slide 16. None of that would be possible, a personalized approach or having a clinical ready Phortress immunophage library, if you can't make your immunophage at clinical scale. So we have our own plant in Maryland. We actually have 2 facilities. We have one in Boston where we do our discovery research and clinical operations. But down in Maryland, we manufacture immunophage, and we do this at the 10-liter scale. Now, in the world of manufacturing, 10 liters is pretty tiny, but because this is using the well-understood principles of microbial fermentation, and because these are bacteriophage, it's incredibly efficient. So from 10 liters, we typically can produce 5,000 to 10,000 clinical GMP-grade doses. Moreover, it's incredibly fast, and we can go from the whiteboard to bulk drug material in about 4 weeks' time. Now, that's what we're doing today. In the future, if we scale up for larger pivotal studies or commercial grade, we can easily move up to 100, 1,000, 2,000 liters. It's very scalable. I'd also point out that we can scale down because if we're doing neoantigens, we don't even need 10 liters. So this is a very flexible system. Marie-Louise?
Thank you, John. So I do have some clinical data to share with you, but first a little bit of background to our study. So currently we're running a phase 2 study with a combination of pembrolizumab and our ASPH phage 301 in patients with locally advanced and metastatic head and neck cancer. We're looking at the safety and the efficacy of the combination. So why did we choose head and neck cancer? Well, the response to PD-1 inhibition is low or moderate, 18%. So that means that more than 80% of the patients do not have an objective response to PD-1 blockade. Furthermore, we know that one of the factors that makes you not respond to PD-1 inhibition is the lack of T cells. And head and neck cancer tumors are known to lack T cells. And without T cells, there is no way that PD-1 blockade can work. So it does make scientific sense to add vaccination, generate and drive the T cells into the tumor to get a better response to PD-1 blockade. Another bonus with choosing head and neck cancer patients is that we want to have tumor tissue. So it's really important for us to look at the tumor tissue before combination treatment and during combination treatment to see that we're doing what we think we're going to do to increase the T cells in the tumors. So let's go to the next slide. I'm in slide 19. So here we have the study design. So we have a combination treatment, but we're mostly interested in evaluating the additive effect of 301 to a PD-1 blockade backbone. So we have a unique design for doing that. So we're taking patients that are not expected to respond to PD-1 blockade alone. The patients we enroll are locally advanced metastatic head and neck cancer. They all had PD-1 blockade for at least 3 months, and only patients with stable disease or progressive disease can be enrolled into our study. With this design, we have a better chance of evaluating the additive effect of 301. So far, we have treated 12 patients with a combination. We have a very nice response already. So we have met the predefined criteria to add patients to the study to a total of 30 patients. So in this first signal-seeking part of the study, we did see some signals. So we have discussed with investigators and also internally in the company, what is the next step? So we want to take our combination earlier in the treatment regimen. So we are going to add a cohort, or actually we already added this cohort, so we can start enrolling patients to cohort 2. We want to treat patients with PD-1 blockade that didn't see PD-1 blockade yet in the first line setting. One very good reason to do that is because there is very nice benchmark data in Keynote 48 and also in Keynote 12. So we can compare to historic data what we are doing with a combination in our patients. We are estimating that we will have enrolled about 30 patients by the end of the year, and we will have the clinical data from the majority of these patients. And based upon this data, we can then make a decision on whether or not to move into a pivotal trial, a randomized phase 2/3. So you heard a little bit about our cocktail approach, and we are very eager to start treating patients with this approach. So we're going to add cohort 3, as you can see on this slide. Approximately in Q3, we hope to dose the first patients with a combination of the current phage, the 301, and a new phage that we just manufactured. So the new phage is an E6/E7 phage. It's targeting the oncogenic drivers of HPV-associated head and neck cancer. So that is going to be very exciting to see. On slide 20, to the right, you can see some immunohistochemical stainings for ASPH, the tumor-associated antigen that we have on our 301 phage. So we are training T cells to recognize and kill cells that express ASPH. So therefore, of course, it's very important that we have a high expression of ASPH on the patient's tumors. So far, we have evaluated 31 patients' tumors, and all of them had a very high expression of ASPH. So let's go to the next slide. On slide 21, you can see the swimmer's plot of the efficacy data from the first 12 patients and 10 patients that have been scanned in the study. So remember that all of these patients had PD-1 blockade before, and none of the patients had an objective response. So only stable diseases and patients with progressive diseases. Another important thing is that the patients were still on PD-1 blockade when we added 301. So there was no interruption of the PD-1 blockade. If we look at these two first patients, patients A and B, received very nice stable diseases for 8 and 10 months respectively. So you may wonder, so what is the meaning of stable disease? Does that really show that the drug is working? Well, if we compare to the Keynote 12 data with a PFS of 2 months and overall survival of 8 months, I think this is very interesting. Durations of 8 and 10 months respectively. If we go down to patient C, we see another very interesting scenario. So this is a patient with PD-L1 blockade for 8 months. He had a stable disease as the best response, but then the tumor started growing. With the growing tumor, we added 301 to his PD-1 blockade, and look at this. We see a nice stabilization of his tumor, and this stabilization lasted for almost half a year. Another important scenario you can see in patient G. So this is a woman with head and neck cancer. She has a PD-L1 negative tumor, as you can see to the left. So because of this, of course, she was not started on PD-1 blockade, because it's known that patients with PD-L1 negative tumors only have a 6% chance of responding to PD-1 blockade. She responded very well to first-line chemotherapy, but then the tumor started growing again. So she was put on PD-1 blockade, and after 3.5 months, she had no response. We added 301, and if we go to the next slide, slide 22, if you look at the graph to the lower left, you can see very nice tumor reduction. So after 6 weeks, this patient had a tumor reduction of 29%, after 12 weeks, 43%, and after more than 6 months of treatment, more than half of her tumor has disappeared, 52%. So this is a clinical response of immunotherapy in a patient with a PD-L1 negative tumor that shouldn't really respond to immunotherapy. As we talked about before, it's really important to look at the tumor. So to the left here, you can see biopsies that were taken before the combination study, and you can see the phenotype of a cold tumor. So at the top, you see very little purple staining, very few CD8 positive T cells. And at the bottom, you can see that this is a PD-L1 negative tumor. After 6 weeks of combination treatment, we took new biopsies. And if you look at the top image to the right, you see a very nice infiltration of CD8 positive T cells, the purple. And at the bottom, you see that the PD-L1 expression has increased, and there is also signs of necrosis. The tumor is dying. So we have converted the tumor from a cold tumor to a much warmer, even hot tumor. We have some additional translational data if you go to the next slide. So here is the gene expression profiling, nanostring, and at the top, you can see a very nice increase in the score for CD8 positive T cells, and most importantly, the cytotoxic T cells that actually are the T cells that kill off the cancer cells. At the bottom, you can see a very nice increase in interferon gamma and granzyme B, which represents an activation of the T cells. So here in this patient, we see that the clinical data with a reduction of tumor burden and also the translational data go hand in hand. So on the next slide, slide 24, we see the safety profile of all the patients treated so far. So as you can see here, the combination was well tolerated. We do see a few grade 1 and grade 2 related adverse events, but it looks well tolerated. Remember though that these patients have been on pembrolizumab for a median of 6 months. So in some ways there could be sort of a selection for patients that are already have shown that they tolerate PD-1 inhibition. But overall, very well tolerated, looking forward to continuing with this study.
So I think it'd be worthwhile to briefly talk about our program in Merkel cell carcinoma. As John said, it's really a microcosm of our future approach to delivering totally personalized cocktail formulations of ImmunoPhage. Merkel cell carcinoma, as you may know, is an aggressive skin cancer, which was recently described to be driven in the majority of patients by a virus like HPV in head and neck cancer. This is the Merkel cell polyoma virus. Now, colleagues of ours at the University of Washington have done a phenomenal job understanding the immunobiology of Merkel cell, and David Cole and Paul Neem have mapped out the patient responses that matter against this oncogenic T antigen of the Merkel cell virus. What we're planning to do is captured on slide 26 in Merkel cell carcinoma is to leverage our ability to manufacture these immunophages. So we've taken all of the antigens that David and Paul described, and we've been able to build 2 phages that express all of those antigenic domains that cover the polyoma virus piece of Merkel cell tumors. But if you look in the box 2 here, we have some preliminary data where we show that Merkel cell cancer patients not only express the virus, but they often express a lot of different tumor-associated antigens. These are those shared tumor-associated antigens that are prevalent across many different tumor types, and we have in our Phortress library. So what we've done is we've just looked to see what kind of frequency might we expect, and that's captured in the upper right here, which is the frequency distribution of this small set of our Phortress antigens. And you can see most Merkel cell carcinoma patients express at least 2 of these, if not more. So what that will enable in the near term is a Merkel cell patient comes in, we do a biopsy, then we do a nanostring analysis with a probe set that reflects our Phortress library. And we could say, oh, this patient is expressing maybe 5, maybe 10 of these shared antigens. And we then put it together with the Merkel cell polyoma expressing immunophage in a totally bespoke cocktail to give that patient kind of the maximal opportunity, the shots on goal, as it were, of the immune system in terms of attacking that patient's specific tumor. So we're really excited about moving forward with this program. So I'd like to pivot real quickly and talk about our VISTA program. This is a recombinant, a human recombinant antibody program. VISTA, as John mentioned, it's in the family of PD-1, both genetically and conceptually. It's a strong T-cell checkpoint molecule. Development of anti-PD-1, anti-VISTA therapeutics has lagged behind PD-1s for a variety of reasons. I think one of the main reasons was for a long time we didn't know what the receptor was. But now we know it, and we had some prior knowledge that helped enable our program sort of get to a jump start. So we do know that VISTA binds to PSGL1, and this has recently been published in the literature as well. Now, another feature of VISTA biology that makes drug development a little harder, and we can leverage our understanding of this to make a best-in-class molecule, is the following. It turns out that VISTA interacts with PSGL1 only in an acidic microenvironment, like in the tumor. In the blood, they don't interact. So if you make, but there's lots of VISTA in the blood. So if you make an antibody that just binds VISTA and blocks that interaction, that would be effective, except that you have issues with PK because the drug gets sopped up. We call it a peripheral sink. It gets sopped up in the blood and doesn't get into the tumor easily. But however, if you can make an antibody that has a similar pH dependence, then you could potentially dose right through that peripheral sink. It doesn't interact with VISTA in the blood, but when the antibody gets in the acidic tumor microenvironment, then you can actually bind your target where it matters. And so we're really excited about moving forward with antibodies that we have that have these characteristics. And these have come from a very successful collaboration that we had with Adimab. And so that project is ongoing.
I'm only going to spend a minute on slide 29 because we're not going to move this program forward in the absence of significant government funding. But we were able to leverage this opportunity to really get some great feedback from the FDA on our program. And more importantly, the cocktail approach that Rob has described is also the clinical trial design that we had proposed to them. We also were able to manufacture 25 unique ImmunoPhage for this program in the space of about 3 months. So we were able to demonstrate our manufacturing prowess. I just want to wrap up the discussion on slide number 30 and point you to some milestones that are ahead for the company. One really important data readout for the company is coming at the end of this year, and that's the phase 2 top-line data readout. Now, that's going to be a rolling data readout, but that opportunity is the first opportunity we'll have to make a go, no-go decision for a pivotal phase 3 study. We're also going to be initiating manufacturing for VISTA and for 401 and initiating IND-enabling studies for those programs this year, and you'll be seeing INDs from the company over the course of the next 18 to 24 months for those programs. All of this is just the tip of the spear, because as Rob said, 401 is just the microcosm of what we plan to do with the platform, and we think we own this space. And right now we're racing to create, manufacture, and patent as many ImmunoPhage as possible, because every ImmunoPhage, every generation of ImmunoPhage is its own business opportunity. We're excited about that. Hope you enjoyed the presentation today, and thanks for your time.